Hello everyone. In the part 2 of this section on the types of fermenters, we are going to study three different types of fermenters. Now in the part 1 of this video, we talked about the stirred tank fermenters, which is largely used for the aerobic organisms. We move ahead and we look at the other fermenters. One of the commonest fermenters that are used is a bubble driven fermenter. There are two types of them, one being the bubble column fermenter and the other is the airlift fermenter. Now the bubble column fermenter, as you can see, is a very simple kind of a fermenter where the mixing is largely done by the upward mobility of the bubbles, movement of the bubbles, as you can see in the picture. And these kinds of fermenters are used for fermentations wherein the organisms used are very sensitive to too much of aeration and agitation. So something like, say, animal cells. In the airlift fermenter, the mixing is largely done by a draft tube. The draft tube can be either an internal draft tube, as you can see in the middle diagram, or the draft tube can be an external draft tube. Now, in case of the internal draft tube, the air is actually passed through the draft tube. So what you will have is you will have a riser inside the draft tube and a downer of liquid outside the draft tube. Now, in case of external draft tubes, the liquid is actually pumped from the top to the bottom. Okay, now what is the function of the draft tube? The draft tube actually prevents the bubbles from coalescing. So smaller the bubbles, air bubbles, larger the surface area, and that will give rise to more oxygen transfer across to the microbes. You can see in the diagram, as I said, the draft tube actually has gas bubbles coming out, giving rise to a riser, and next to it you will have air bubbles going down, the liquid going down, which gives rise to a thorough mixing of the medium. The other type of fermenters are fixed bed bioreactors. Fixed bed bioreactors is almost like solid state kind of a fermentation where the cells are immobilized onto a support. There are different kinds of supports used, like you can have alginate, you can have beads, you can have plastic box, you can have concretes, wood shavings. Like the wood shavings are the very common ones that are used in vinegar fermentation, or plastic or glass wool. So the solid support is actually stacked on top of each other, and you have the medium that is dripped onto it, and the organisms grow on the solid support. As I said, there are different kinds again in this, variations of this, like the one which is used in sewage treatment called as a trickling bed fermenters. The one which is used in production of vinegar. Commonly, these kind of fermenters are used if the organism in question is a fungus. It becomes very easy for the fungus to actually grow around the solid support, and all you have to do is keep dripping the nutrient medium on it, and what comes out through the other side will be having a very high concentration of the end products, commonly used for production of various kinds of organic acids and enzymes produced by fungus and so on. As I said, the industrial applications, it can be production of enzymes, amino acids, wastewater treatment, and so on. The next kind of bioreactors that we're going to talk about are the fluidized bed bioreactors. So this kind of a modification of the first two reactors that we have seen, that is, we have solid supports. We have cells growing on the solid supports, like beads and so on. But then these beads are actually made to move around in the medium, allowing certain amount of mixing inside the medium. So the disadvantage that we see in a fixed bed reactor, where at certain parts of the fixed bed reactor can have deprivation of oxygen, that is kind of negated out in the fluidized bed reactor. Fluidized bed reactor, very, very commonly used to make animal cell products like growth hormones, insulin, and so on. Thank you.